Welcome back friends, welcome back in another video tutorial for microbiology with Shagnik. Welcome in another video of cell biology. So we have talked about how the proteins, which are membrane proteins synthesized in the rough endoplasmic reticulum, targeted to the ear lumen, right? Now in this today's video we will going to learn how from ear lumen it will be transferred to the Golgi, and from then Golgi it will be transferred to the endosome or lysosome or any other thing. Okay, so let's get started with the video. So first we will going to see a story that will help you to understand how the entire thing is going on. Then we will going to talk about various type of important things that we have to learn and we have to remember for answering any kind of question from that particular part. So let's get started with the video. So this is our endoplasmic reticulum, and up to the previous video we have already learned how the proteins which are synthesizing in the RER are coming to the ER domain, right? So if you have not watched this video, so I will recommend you to watch this video and then come back here. Then it will be very easier for you for understanding. Okay, so now in the ER lumen, first of all, remember there are actually two type of proteins that are present. Okay, one is resident proteins, another one is export proteins. So resident proteins are proteins that are mainly involved in in correcting the misfolding of export proteins and in many other processes. Okay, and these resident proteins are actually, as its name suggests, remain in the ER lumen. Okay, they will not going to be exported or transferred, right? And the other type of proteins, which is export protein, will going to be transferred. Okay, so one thing is clear from now, that export proteins will going to be transferred. Okay, and the other type of protein, which was the resident protein, will going to maintain the export protein, right, in the ER. So now how the we will going to answer how this export protein will going to be transferred, right? So this export protein will going to be transferred with the help of vesicles, which are known as transport vesicles. Okay, these transport vesicles will going to transfer those export proteins from ER to the cis Golgi, okay, which is the first compartment of the Golgi complex. Okay, now remember, from this side the first Golgi will be known as cis Golgi, and from this side the first Golgi will be known as trans Golgi. Okay, so now there will be transport, or there will be formation of transport vesicles, which you may think like a car, and inside that car there will be our proteins. That proteins will be transferred to that Golgi. Okay, now the transport will not only be in this direction but also in this direction. Okay, we will going to transfer what kind of protein are going to transport from ER to Golgi, and what kind of protein are transport from Golgi to ER. But first we have to learn how these cars are forming, or how these membrane vesicles are forming. Okay, so as you can see, these vesicles are actually forming from the membrane of the ER. Okay, so in the membrane there will be specialized coated region from where these vesicles will be present, or from where this vesicle will be formed. Okay, and this specialized region of the membrane will be covered with proteins at its cytosolic end, so that it can easily bud off. Okay, now once they have bud out with the protein inside, they will go for the destination. Okay, and before they will be transferred or they will reach the destination, the proteins which are covering that vesicles should be removed. Okay, so that that particular vesicle can fuse with that particular recipient membrane. Okay, so that is the thing. So what will happen first, from the region of the membrane which is coated with proteins, especially at the cytosolic side, there will be formation of vesicles. And now after the formation of vesicles, the vesicle will be transferred to the destination, and once the destination is reached, just before that, the proteins which are covering that vesicles will be removed. Okay, so that that particular vesicle can fuse with that cis Golgi network. Okay, although this is the cis Golgi network, and you can also remember that before going to cis Golgi, this is another station where that particular vesicle can fuse. Okay, and then from here there will be another vesicle that will form, and it will going to be transferred to the cis Golgi. That's how the thing is going on. Okay, so that's the idea, overall idea. Now the question is how these vesicles are forming. So these vesicles are forming by reversible assembly of proteins. Okay, and in our body there are three type of vesicles that have known, and we will going to discuss, and these three type of vesicles are formed from different type of proteins. We will going to see three type of vesicles. One vesicles is formed of COP1 protein, another vesicle is formed of COP2 protein, and another vesicle we will going to see which will formed of Clathrine protein. Okay, so now the question is, as these vesicles are named according to the proteins from which it is made up of, does that mean that this particular vesicles is only composed of that particular protein? So the answer is no, because not only those COP1, COP2 or Clathrine protein, along with that there will be some adapter proteins which are also very very important, as well as there will be some GTPase proteins or GTP binding proteins that will be present. Okay, so the main proteins along with adapter proteins and some small GTP binding protein together reversibly assemble to form the membrane vesicles, which will going to play the role of a car to transport protein from here to first vesicular tubular cluster, then from there cis Golgi, and from there this part of the Golgi, then here, and that's how the thing will going, right? So here first we will going to see different type of vesicles, how they are forming and what are the role, because you can see here the COP2 coated vesicles which are transferring protein from ear to the Golgi, right? And here it is COP1 coated vesicles which are transferring protein from Golgi to the ear, right? So it is obvious that different type of protein coated vesicles have different job, right? So let's see what types of coated vesicles are present and what are their job. So here we will going to see first about Clathrine coated vesicles. Now these Clathrine coated vesicles have many subtypes depending on with which type of adapter protein it have assembled. If that Clathrine protein have assembled with AP1 or AP3 adapter protein, the vesicle will be formed from trans-Golgi network and it will be transferred to the lysosome. Okay, and if that Clathrine protein will going to assemble with AP2, the vesicle will be formed from plasma membrane during the endocytosis and it will transport to the early endosome. Okay, so that is the case in case of Clathrine. So if we are going to see that if the Clathrine is attached with AP1 adapter protein, it will going to transfer protein from Golgi complex to the endosome, right? And if it will going to assemble with AP2, then it will transfer the proteins from plasma membrane to the endosome. Okay, so in both the cases it is going in this direction. Okay, so that is the thing in case of Clathrine. Now let's talk about COP1 coated vesicles. So remember there are two type of transport present. One is retrograde transport, another one is anterograde transport. Okay, so retrograde transport is the this type of transport. Okay, where protein are transferring from Golgi to the ER, and anterograde transport is this type of transport where protein are transferring from ER to the Golgi. Okay, so first type of COP coated vesicles that we are going to see is COP1 coated vesicles, and they are responsible for retrograde transport. Okay, that means transfer of protein from Golgi to ER. Retrograde transport are also known as intra-Golgi transport. Okay, because intra-Golgi transport are also similar thing. Let's say transfer of protein from trans-Golgi to the cis-Golgi. It is an example of intra-Golgi transport, right? So that is the idea. So here we will going to see how these COP1 coated vesicles are activating. So it all starts with GEF protein GA1 or GA2. This GA1 or GE2. So before going to talk about this, I want to talk with you about some G protein activators as well as inhibitors. So there are two things that we will going to talk about, and you will going to learn about these things in details when we will going to talk about cell signaling. So for now, you have to learn only those things that I am going to tell to you. Okay. So first type of protein, that is GEF. GEF means, remember, guanine nucleotide exchange factor. Okay. So remember, this type of protein will going to give GTP to any protein. Okay. And another type of protein that is present are known as GAP. GAP means GTPase activating protein. And remember, if any proteins are attached with GTP, this GAP has the ability to remove that GTP from that particular protein and attach GDP as a replacement of GTP. Okay, so this is the two type of thing we should remember to understand how these vesicles are forming. One is GAF, another one is GAP. And they are the topics of G protein coupled receptors. You will going to learn this when you will going to study the cell signaling. Okay, so let's come back to the topic. We are talking about how COP1 coated vesicles are formed. So there are two type of GAFs that are present. One is GA1 or GA2. So GAF means it will going to give GTP to any kind of protein. So GA1 or GE2 will going to activate a GTPase activating protein or a small GTPase protein named ARF1. Okay, now this small GTPase ARF1 will going to recruit ACC21, ACC26, RAD2 and RAD3. And these four things together will going to form the inner coat of the COP1. Understood? So that is the idea. So again, if I going to repeat this thing, so there are redundant gaps. Gap means again it will going to provide GTP. So these gaps are GEA1 or GEA2. This GEA1 or GE2 will going to activate small GTPase protein named ARF1. Now this ARF1 thing will going to in turn recruit various type of things that going to play an important role in the inner coat complex formation, SEC21, ACC26, then RAD2 and RAD3. Okay. Along with that, another SEC proteins, for example, SEC27, SEC28 and one RAD protein named RAD1 will going to form the outer coat, and they will together assemble to form a triskelion structure, that is COP1. Okay. So now the formation of COP1 can be inhibited with the help of Brefeldin A named lactone antibiotic. And how they works? They works by inhibiting the GAFs GA1 and GE2. Okay, so that is the idea of how COP1 coated vesicles are formed. Okay, so now let's move to the next type of vesicles, that is COP2 coated vesicles. And now if we going back to the diagram, COP2 coated vesicles are responsible for enterogate transport. Enterogate transport means, so COP2 coated vesicles are responsible for enterogate transport, that is the transfer from ER to Golgi. So now let's talk about what are the COP2 coated vesicles. So as I have told you, it is responsible for enterogate transport, that is the transfer from ER to the Golgi. So now let's talk about how they are activating. So it all starts with the ER membrane bound GIF, that is Sec12. This Sec12 will going to activate small GTPase named SAR1. Okay, so this SAR1 will going to in turn recruit a dimer Sec23, Sec24. Okay, which will going to play important role in the formation of inner coat. And then this Sec24 and Sec23 recruitment, there will be recruitment of Sec13 and Sec31 that will going to create the outer coat. And then polymerization will occur that will create a lattice-like structure that drives the membrane curvature. So that is the idea of COP2 coated vesicles. Okay, so three type of vesicles we have learned. One is Clathrine. The job of Clathrine is depending upon which type of adapter protein it have assembled with. If it have assembled ADP1 or ADP3, then remember the vesicles that is formed will going to act as a transport vesicles from trans-Golgi to the lysosome. Now if that Clathrine have combined with ADP2, then it will going to act as a transport vesicles from plasma membrane to the endosome, right? And then we have talked about COP1 coated vesicles. COP1 coated vesicles, remember, are important for retrograde transport or intra-Golgi transport, that is the transport from Golgi to the ER, right? And here we have talked about COP2 coated vesicles that are responsible for enterogate transport. So that's the idea. So I hope you have learned about the three type of vesicles very well. Now in the next part of the video, or which we will going to talk about in this video also, I am just talking about the next topic, that is a question. That question is, in the beginning of that video I have told you that there are two type of proteins that is present in the ER domain. One is responsible for your export proteins, one is export protein, another one is resident protein, right? So the question is how the transport vesicles as well as how the cell recognize export proteins from resident proteins and transfer them from ER to Golgi instead of those resident proteins. And if somehow that resident proteins by mistake transferred to the Golgi, then how they can be retrieved. Okay, so idea is, in case of the ER resident protein, there is a special type of sequence present, especially at the C terminal with that protein. This sequence is KDEL sequence. Okay, this KDEL sequence, this KDEL is for the short, not short, that is the one letter abbreviation of amino acids. So K means here your lysine, D means aspartic acid, E means glutamic acid, and L means leucine. So this 4 amino acid containing sequence is present in case of resident proteins. Okay, so that is the reason why these resident proteins are retained in the UL domain, and if they somehow by mistake transferred to the cis Golgi, they will be retrieved back. Okay, and obviously in both cis Golgi as well as endoplasmic reticulum lumen, there will be presence of some KDEL receptor. Okay, this KDEL receptor will going to recognize and bind those proteins having KDEL sequence at the C terminus. Okay, so that they can be retrieved back to the ER lumen. Okay, so that's the idea. Now, what are the evidences that supports this theory? So there are various experiments that have done by the scientists that supports this theory. For example, they have removed that KDL sequence from the resident proteins, and with the help of recombinant DNA technology, they have added the KDL sequence in the export proteins. And what they have seen? They have seen that those export proteins are retained in the ER lumen, and the resident proteins are transferred by the vesicles we have just talked about. So it is amazing, right? So that have supported the theory that this KDL sequence is enough for recognition of the resident proteins and their retrieval from cis Golgi network to the ER, right? So that's the idea. So not only that, for the transmembrane proteins, there are special type of sequence present having two lysine residues at the C terminal. Okay, so that's the idea of how resident proteins are retained in the ER lumen, and if they are somehow transported to the cis Golgi, then how they can be retrieved. Now, another important interesting thing that the affinity of the KDL receptor for the KDL sequence is higher for cis Golgi as well as for the vesicles present. Okay, because the pH of the vesicles and cis Golgi is lower than the pH of the endoplasmic reticulum. Okay, and it is thought that KDL receptor has higher affinity for that KDL sequence at low pH. Okay, so that's the idea. So that's the video for our today's discussion. Today we have learned how from the ER lumen the proteins can be transferred to the Golgi. Okay, so I hope this video will be helpful for you. Thank you.